document for applying
for
for
genome edited
genome-edited
product
product
as a
as a
variety.
variety.
Okay,
Okay,
Dr.
Dr.
Santosh, please.
Santosh, please.
So very
So
good,
very
good
good
afternoon to
afternoon
all.
to all.
So my
So
name
my
is
name is
Santosh.
Santosh.
I'm
I'm
currently I'm
currently
working as
working
research
as research
associate
associate
with
with
Dr.
Dr.
Vishwaradhan
Vishwaradhan
Chinnaswamy,
Chinnaswamy,
Joint Director
Joint
at
Director at
IRI.
IRI.
So
So
today
today
I'm
I'm
talking
talking
about
about
how to
how to
prepare
prepare
a
a
standard
standard
SOP
SOP
document.
document.
For
For
example,
example,
if
if
anybody
anybody
has
has
any
any
genome-edited
genome-edited
line with
line with
TDNA
TDNA
free,
free,
so how
so how
to
to
go
go
forward.
forward.
So
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 earlier,
earlier,
we know
we
that
know
the
that the
transgenic
transgenic
research,
research,
so it
so
is
it is
regulated
regulated
under the
under the
Government of
Government
India
of India
1989
1989
rules.
rules.
So,
So
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 earlier,
earlier,
so whatever the
whatever the
GM
GM
crop which
crop which
harbors
harbors
TDNA.
TDNA.
So
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 generally,
generally,
recently
recently
the
the
mutants, for
mutants, for
example, the
example, the
mutants
mutants
developed by
developed by
SDN1 and
SDN1 and
SDN2
SDN2
approach.
approach.
I
I
don't
don't
discuss
discuss
all about
all
the
about the
details
details
since
since
I think
I think for
for
last for
past
past
classes, I think
classes,
you
I think
have
you
heard
have
all
heard all
about this.
about this.
What is
What
guide
is guide
RNA,
RNA,
TDNA,
TDNA,
and
and
genes how to
genes how to
select.
select?
I
I
think
think
all you
all you
understood.
understood.
So now
So now
only the
only the
thing,
thing,
just
just
I'm
I'm
telling
telling
just
just
the
the
basics of
basics of
what are the
what are the
things we
things
have to
we have to
consider for
consider for
the
the
preparing the
preparing the
document.
document.
For
For
example, the
example, the
mutant
mutant
developed by
developed
anybody,
anybody,
those
those
anybody
anybody
developed by the
developed by the
mutations
mutations
by
by
using
using
SDN1
SDN1
or
or
SDN2
SDN2
approach.
approach.
So
So
generally,
generally,
the first
the first
generation, for
generation, for
example,
example,
the
the
plant will
plant will
have
have
transgene.
transgene.
So
So
transgene
transgene
means it
means it
has, for
has, for
example,
example,
guide
guide
RNA,
RNA,
Cas9 and
Cas9 and
selection marker,
selection
it
marker, it
occurs
occurs
within the
within the
TDNA
TDNA
region.
region.
For
For
example,
example,
you
you
got
got
mutation
mutation
in this
in this
in the first
in the first
generation
generation
with
with
desirable
desirable
mutation.
mutation.
It could be
It could be
considered as
considered
a
as a
transgenic crop.
transgenic crop.
So in the
So in
second
second
generation,
generation,
so
so
plants with
plants with
desired
desired
mutation
mutation
in the
in the
segregation, in the
segregation, in the
Mendelian
Mendelian
segregation
segregation
ratio,
ratio,
it's
is
segregated.
segregated.
So
So
here you
here
will
you
get
will get
the
the
Cas9
Cas9
positive
positive
and
and
as well
as
as
well as
Cas9
Cas9
negative
negative
with
with
desirable
desirable
mutation.
mutation.
So
So
here
here,
uh,
uh,
in this
in this
case,
case, we
the
the
whatever the
whatever the
mutation, you are
mutation, you are
indistinguishable
indistinguishable
from
from
natural
natural
and
and
normal.
normal.
So it is
So it
free
is
from
free from
transgene.
transgene.
So
So
this could be
this could be
considered as
considered
a
as a
non-GM,
non-GM,
non-genetically
non-genetically
modified
modified
plants or
plants or
non-living
non-living
modified
modified
organisms.
organisms.
So
So
hence,
hence,
so
so
genome-edited
genome-edited
crops do not
crops
elicit
not elicit
any
any
biosafety
biosafety
and
and
environmental
environmental
safety
safety
concerns.
concerns.
Means it
Means it
does not
does
have
not have
any
any
TDNA.
TDNA.
So several
So several
countries
countries
also
also
recently
recently
like they
they
used
used
USA,
USA,
Japan, and Israel, Argentina, and Brazil, Chile. Some of the countries, they are considered genome editing crops free from foreign gene as a non-GM crop. So to develop globally competitive Indian Agriculture Research Institute and ICAR, so the government of India, the government of India, in harmony with global regulation of genome-edited crops, they made a decision on 30th March 2022 to exempt genome-edited plants of SDN1 and SDN2 category free of introduced exogenous DNA from GM regulation. So this is the guidelines. So before starting, if anybody wants to start the genome editing, want to do any genome editing, first make sure that, so after designing your guide RNAs and vectors, after deciding, before proceeding, you have to submit all the necessary things to IBSC, Institution Biosafety Committee. First, you have to make a document. For example, you have to submit, you have to go to this Indian Biosafety Knowledge Portal. There you have to submit the form D1 to form D1, which has all the necessary things. For example, selection of gene, guide RNA details, plasmid constructs, and so in which facilities you have to mention the where you have to perform the, for example, if you get any mutant in future, so where to perform all the experiments and related, and so all the things you have to submit to IBSC. So once in IBSC meeting, they approve, they will screen what are the details which you have given. Then after that, the IBSC which is sent to RCGM. So RCGM we again will screen, and they will give one unique UAC number, unique accession number, unique number they will give. So that one you have to keep a record. So once, so as I told, this is a transgenic. So these are the things you have to mention. sgRNA design agrobacterium. So what type of means method, agrobacterium or gel, any particle bombardment or any protoplast, you have to give the mention the details. And so how to recover the edited lines, how to characterize the mutant phenotype, and so production of transgene free. So since it harbors a transgene, so these are regulated under 1989. So as I told, so once if it passes through, once you have the mutant line, then how to proceed further. So for example, this is a field testing. So how to do field testing in the in future. So for that one, Government of India, they have given some standard operating procedures. They have given. So what are the things you have to take in the consideration. So so these are the protocols to show the genome-edited plants are free from exogenous introduced DNA. So you have to mention, as I told, genome editing. So which method, either CPF1 or base editing or Cas9. So you have to mention the construction of vector for editing of desired genome of interest, and also the method of translation procedure in detail. So which method, if you modified everything, you have to mention. And also, for example, if you get mutation, you have to give the nucleotide sequence, parental, which genotype or any parental line, you have to mention which mutant and corresponding mutant in which parent you have developed desired gene, and you have to give sgRNA details. And also you have to give the nucleotide and protein sequence alignment. Also you have to mention between parent and wild type, and mutant line, it's a Cas9 if it is a Cas9 free or transgene free. And the other one is the absence of selection or scoreable marker, and you have to give the phenotypic information of desired mutant. And the last but one, so complete vector sequence. So which vector sequence which you have cloned all your guide RNAs, the all information you have to mention. And what are the primers which are used for nested PCR or overlapping PCR. So this is a very important PCR. So now I'll explain. So according to this standard, before you are making dossier, so you have to design primers from all the total the vector which you have used. For example, the vector is 15 kb. You have to run, you have to design primers consecutively. Make sure that the primers which cover whole the vector. So I am showing one of the example. So since I am working on one of the gene in rice, it's a drought and salt tolerant gene. It's a zinc finger transcription factor. So it's a negative regulator which enhance it which enhance drought and salt tolerance. We made so we have some Cas9 freelance in this background with desirable mutation. So as I told, so 2017 we earlier already submitted this to IBSC for proposing. After that we made transgenic line. Again, once you fulfill, once you get the mutation, again you have to make a document like this. This is as like I told, you have to submit to IBSC to this document, and IBSC will approve, and again IBSC will send to RCGM and all the things, etc. So this we approved by this one recently. What are the dossier which we have prepared? So in IBSC 12th one, it was accepted, and we have presented in 258 RCGM meeting, and finally it is approved in 259 RCGM meeting on 31st May 2023. So I am telling the example. So this is the dossier. You have to mention the name of the investigator or like the one who developed, and you have to give the details of as I told USC number. So you have to give the USC number which you have submitted earlier. So again, you have to mention the name of the institution and chairperson and member any secretary. See, this is important. So details of the genome-edited line. For example, which plant species you have used. So what are the target sequence and what is the name of the genome-edited line. So which you have mentioned. Whether it is category of SDN1 or SDN2. And again, you have to mention about, as I told, target genomic region. You have to mention everything. Since here not possible to, it is not possible to incorporate all this in in this area, and this here you have to give as a through annexure. So you have to give the complete details of your gene, and also you have to mention the which method you use for CRISPR, whether it is CRISPR-Cas or TALN or ZFN and whatever. And details of the vector, you have to mention the details of the vector and the method which is used for delivery. Since we have developed several methods for transformation, so here we develop through agrobacterium mediated transformation in rice. And also you have to mention the current generation. Means, for example, if you get mutation in T0, if it is heterozygous, again from starting onwards till the Cas9 free plant, so where you got, so every information with sequence and as the chromatogram and alignment of the gene, everything you have to mention. So, and the next one, you have to, so you have to give the phenotypic data which you have any, you have to mention the phenotypic data, any, so so like that you have to give all the information. So this is one of the line which I got in Cas9 free in T2 generation. So this is the line I have, for example 8-5-6. So this is the line. So from this I got Cas9 free line. So this line I further I processed. So as I told before, before proceeding nested PCR and all the things, so you have to prove that so the your genome edited free line which is which it has absence of selection marker. Here in this vector I used selection marker HPT2, hygromycin phosphotransferase 2. That's why the background, the parent which I have used is MTU 1010. It's a mega variety. So this is the line of DST line which I got Cas9 positive. So as I told, from this line I got in T2 generation Cas9 negative. So what I did, I grown all these three lines in the same in half MS media as well as half MS media with hygromycin. See, so this is for 5 days after germination and this is after 10 days of, this is 10 days after germination. If you see clearly, this is all the seeds were growing, grew very well under normal half MS media condition, and this is for after 10 days. If you see here, so after 10 days the MTU 1010 lines and the Cas9 free lines are not growing, only the parent which Cas9 positive, it is growing healthy in hygromycin media. It clarifies that, so these lines which are free of hygromycin selection. And is there for instance I have given one example. So the mutant which has more broader leaf width and also flag leaf area, this is one of the phenotype. I mentioned only one. Several phenotypic characters were also there in DST mutant, which increases yield and as well as for stress tolerance also it was there. So for preparing dossier, this is the one of the important parameter. So overlapping or nested PCR. So this is very tricky and very important aspect in the preparation of dossier. So please listen carefully. Why? Because any doubts, just let me know. So this is my vector map. So my vector map is PKM 1300 having I cloned 2 guide RNAs. Okay. So this is my vector. So my vector size is 15,114 base pair. So I made, since as SOP procedures, you have to cover all the vectors. So they mentioned all the vectors. So each amplicon, they mentioned each amplicon. I'll explain. So each amplicon, it should be at least 500 bases. It is even 501 also they won't accept. Only it should be greater than or equal to 500. It should not be it is even 501 502 also. So make sure that it is very important. And no sir, it is exactly 500. Even 501 also they won't accept. Only exactly 500. Less than 500 is accepted. So it is greater than or equal to 500. 500. Sorry, less than or equal to 500. Sorry, less than or equal to 500. Yeah. So, for example, this is exam almost all 500 nearer. And one more they mentioned, two consecutive amplicons. The overlapping distance would be at least 50 bases. At least the overlapping distance they mentioned it should be 50 bases. Yeah. I'll explain, sir. That is also like that. I made total of 52 amplicons from C. I started from right border. So right border P1. So these are the like 1, 2, 3. So like that 44 again C. Since it is a raw circular, so this is the overlapping here at the last. Okay. So these are the primers just which we made. So this is a very tough task to make for to cover the all the to cover the vector. So before running the overlapping PCR, so after first you have to isolate good quality of DNA and make sure that you have to check whether the DNA is working. We don't know since after it's a PCR, we make sure that your DNA is working properly or not. For that one you have to choose endogenous any low copy number. So all this they have given guidelines in SOP. So a well known endogenous low copy either like actin, tubulin of the same species using the same amount of the same genomic DNA sample of the final edited line as mentioned above. And the genomic DNA of segregant line means the Cas9 whatever line harboring full or part of the exogenous introduced DNA using the same primer. You have to run the same primer with positive line as well as negative line. Okay. And wild type as well. And also the genomic DNA of the parental, as I told here, as a positive control. So earlier also we think that generally we use positive control as a plasmid we used to run generally. But here they mentioned wild type control, wild type as a negative control. So for positive control they mentioned whatever the parental genotype, you have to spike. You have to spike with your plasmid DNA with the ratio of almost 1:10,000. Okay. Of the full length purified vector DNA used to develop the genome edited line using the same primer set for overlapping PCR. Okay. And the other thing is the amount of template DNA and PCR condition should be such that clear band of expected size is visible. It is not like that. So the clarity of visible band, it should be very fine and proper. It's not like faint. They won't accept all these things also. So make sure that if you get any faint and all the things, make sure that your DNA, it should be, at least you have to recheck your DNA and all the things. Okay. Or the conditions which you have, since the primer to primer, the conditions, it may vary or it might be same. And the one most important point is no amplification should be detected in any of the reactions with primers directed against exogenous introduced DNA for the final genome edited line, while clear amplification should be detected in all positive controls. As I mentioned, any your parent wild type DNA and as a positive control wild type with spiked with your purified plasmid DNA. Okay. Anyway, I'll show it to the gel picture, then you will understand very clearly. Okay. As I told, so for example, this is my positive, this is my forward primer. See, this is just for example, then how I made the overlapping. So this is my forward primer So the red one is reverse primer. So this is forward primer and this is reverse primer. And the overlapping between, for example, this is one amplicon and this is another amplicon, and the amplicon distance, the overlapping distance, you have to count from here to here. Okay, so the overlapping distance should be at least 50 bases. So like that you have to make. Understood or not? Okay, so like that I have shown here. So see, for example, the first one amplicon, my size is 497 base pairs, and second one is 445 base pairs, and third one is 445 base pairs. As I told, like the overlap distance, it is more than 550 base pairs. See, so this is the overlapping distance, and between P2 and P3 consecutive amplicons, the distance is 365. This is also not at all a problem. So like that, it is you have to mention, you have to give every detail in your SOP. As I told, so after isolating DNA, what I did, so first I'll tell you. So wild type, my parent genotype. So I spiked with parent genotype with my vector DNA. So this is my mutant line with tDNA, I told. So this is my line, final genomated line with Cas9 free. So I isolated DNA and I purified. So first I tested my genomic DNA, all the DNA with internal control and DST also. Why? Because since DST also, it is a single gene in the rice genome. That's why I used. So this is my mutated sequence. This is my mutated DNA. Since disruption of this DST mutant leads to 366 base pair deletion, it was happened. That's why this is also DST. Don't confuse. So this is wild type amplicon. This is wild type with spike amplicon, and this is my Cas9 positive plant with desirable mutation, and this is my Cas9 negative plant with desirable mutation. Okay. So I tested with actin and the DST. See what they mentioned. The amplicon should be very clear and fine. Okay. So after checking, so then as I told, I made almost all 52 different type of primers, and each primer with these 4 reactions. So these 4 samples, one is with wild type and spike, and this one. So each one so this is wild type, and this is wild type with spike, and this is with my positive Cas9 positive, and this is Cas9 negative. I think you should ask why you are not getting here. Since my tDNA, this is right border, so it is integrated within the genome. So during this transgene integration, so right border it was not there in this positive line itself. So this is my negative, and where it was integrated in my genome, this is the right border. After right border, see again. So my next second one, wild type, this is positive, and with positive, and so my line which has negative. Again like 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, sorry, okay, 12, 13, 14, 15, 16, 17, 18. See here 19, 20, you see extra bands you are appearing here. You should ask, it is more than 500 bases. You should ask why again, as they are mentioning about 500 bases, and why again you are showing here it is more than 500 bases. The reason is that, yeah, so here, so whatever I have, see if you observe here, I put some sequences like some nomenclature, add 1, add 2, add 3. Why? Because in that region, so the hygromycin is driven by 2x35 promoter. It means that the whatever the 30s5 promoter sequence, it is running as twice. If it is running almost all 300 to 400 bases, so it is very difficult to make a primer with repeated sequences. So for that time, we also don't know like how to solve this problem. Finally, we resolved and we made again. So whether really it is due to because of the repeated sequences or not, then again we designed some primers, and again we have so here that's why here I am getting. So to confirm these whether these sequences whether really it is positive or any unwanted bands, it should not be any non-specific band. Okay, so that's why to test this, to confirm these sequences, again we run with the additional primers that so these are the true original sequences of vector DNA. Okay, so like that it is see here it is 25 and 26c. So 26, yeah. From here onwards again you are not getting any band in Cas9 positive. Why? Because so this is the region of left border. So the first one is right border, and second one here it is the 26th one is left border. So the in transformation what happened? So right border and total whole tDNA was integrated within the genome. That's why you are getting you are not getting band after this out of the left border. It means that only the tDNA border it was integrated, the backbone it was not integrated within the genome. Okay, so this is like that. If you see 26, so there is only you got band only in the wild type with spike. Why? Because the spike means the vector DNA or plasmid DNA. If you add with wild type with plasmid DNA, since it harbors the plasmid DNA, obviously you will get the clear band in this wild type spike background. So like that we fulfill all the amplicons till 44. Okay, so currently these lines, so now these are under trials. So probably maybe 2000, maybe 25, 26, maybe it may come. So currently lines are testing for, so we are giving testing for yield as well as drought and salt. So I thank to the organizers of NIPB and NAHEP for giving me this opportunity, and also my sir Dr. Vishwanathan, and he has supported me to do this work, and Dr. S. Gopal Krishnan from Division of Genetics, he assisted me in like for analyzing experiments, and he is given some suggestion. And these Dr. Sohamre, Dr. Shivani Nagar, and Pranjal Yadav, Anchana Bhatt, these are the scientists I also thank. Why? Because these are associated with Vishwanathan sir in our lab. And also I thank my lab members and also Division of Plant Physiology, and thank you. So if you have any doubts.
सर ये जो SOPs आप बता रहे हो कि ये जो NX है जो भी फॉर्म फिल करते हैं वो ये जो रिसर्च ऑर्गेनाइज करने से पहले या बाद में करते हैं?
जो मैं पहले वाला में नहीं दिखाया, पहले वाला जब when I was starting in 2016, so before, for example, if some exchange you are going to do, that time you have to submit to IPSC, your institute. So your institute will review it, and it will go to RCGM, and RCGM.
But sir, as you told that this transformation method, how we will know that transformation method only will work in our whatever we decided? Because
no,
so many
that's what optional. You have to give the earlier. You have to give before doing experiments. You have to give the all transformation methods like slash. You have agrobacterium or slash. You have to give.
Okay.
Once if you develop the line, since you develop line through some method. So after that, that dossier you have to mention. Initially you have to give some option there. But once you get the line there, you must have to give which method you used.
Okay.
Okay. But that's why I told UAC number they will give first. That number indicates that so you're allowing to work for this genome editing work for next for further. So any gene, if you want to do work, you have to submit IBSC form. कोई भी है तो हो गया ठीक है ठीक है
Okay, so we thank you, Dr. Santosh. Dr. Santosh.